So here is the missing information from the recording on Friday, December 4th. So we're picking up with the last part of Mendel's laws of segregation. So we were talking about on Wednesday that alleles don't blend, and so they're segregated during meiosis into the gametes in the parents. And then when they undergo fertilization, the new organism, the offspring, those alleles aren't necessarily going to be expressed in that organism. So heterozygotes, remember, those are the organisms that inherit two different alleles, two different copies of the gene. So heterozygotes are only going to express the dominant allele. So what happens then to the recessive? The recessive allele is going to basically end up being hidden. It's going to get covered by the dominant allele. They'll still have the genetic information, but it won't get expressed because the expression of the dominant allele is going to get covered, or is going to cover up that recessive information. So then more on Mendel's observations. So this time, instead of looking at flower color, we're looking at peas themselves, the pea color in this case. So looking at yellow and green. So we're looking at the P0 generation, the parental generation, and we've got true breeding strains again with yellow peas versus green peas, green being recessive. And so those parental generations pass on their genes in the gametes to get us the F1 generation that is heterozygous. And in this case, we've got yellow is the dominant, so we do have the yellow and the green gene, but the yellow is the expressed gene. Then that F1 generation, remember in his experiment, he allowed it to reproduce by self-fertilization, and so it will pass on in its gametes either the dominant gene or the recessive gene. So then we get the F2 generation, and the F2 generation, we get either yellow or green offspring. But what we can see this time is the actual genetic combinations drawn out in a Punnett Square. And so we get the two dominants genes, so homozygous dominant, heterozygous offspring, all of which have the yellow seeds, and then we get one homozygous recessive, which will have the green genes. So with the 3 yellow and 1 green, that gets us that 3-to-1 ratio that we saw before. So you can see that 3-to-1 ratio down here. But this gives us some new information and what we call genotypes. So the genotype is the genetic makeup of an organism. And so here we're talking about the alleles that are inherited. So that's what we mean when we talk about homozygous dominant, heterozygous, or homozygous recessive. And so if you are heterozygous, that is your genotype. And your genotypes in this F2 generation from true breeding parents in the parental generation is always going to come out to roughly 1 to 2 to 1. Because if we look here in our Punnett Square, we get 1 homozygous dominant, so 1, 2 heterozygous, 2, and then 1 homozygous recessive. And so we get 2 different ratios, 1 ratio for the genotype. So in this case, it's 1 to 1. So from that true breeding strain of the parents, we're going to get in a monohybrid cross a 1 to 1 ratio. And then you get the phenotype. And the phenotype is the physical expression of those genes. So in this case, seed color, or in the case of the flowers, if they're purple or white. And so the phenotype is the ratio that we talked about before, the 3 to 1 ratio. So 3 yellow phenotype to 1 green. So then the question is, can you tell an organism's genotype from its phenotype? And we talked on Friday about this, and the answer is no. You can't always tell the genotype from the phenotype. You can for the recessive, because you have to inherit 2 recessive genes in order to see the recessive phenotype. It's the only way you can see the recessive phenotype in a dominant recessive gene in organisms. But you cannot tell the genotype for the dominant phenotype, because the dominant phenotype could have the genotype of homozygous dominant or heterozygous. And so there's no easy way to just look at an organism and tell its genotype for an organism that has the dominant phenotype.